All right, so we have entered chapter 5, and we're going to go over in this mini lesson the beginning of chapter 5, which is the cell membrane and the membrane structure. So let's go ahead and start talking about the plasma membrane. So we already know that the plasma membrane keeps the inside of the cell inside and the outside of the cell outside. So it creates a barrier. But this barrier is selectively permeable. So that means that certain things can enter the cell more easily than other things. So we're going to talk about this selective permeability that our plasma membranes have. So let's do a little bit of review. Back in chapter 3, we talked about the phospholipid bilayer. So if we take a look at this phospholipid bilayer, we know that there is the phospholipid part, or the phospho part, I should say, is going to be hydrophilic or water-loving, and then the tails are going to be hydrophobic or water-fearing. So this bilayer that we have has the hydrophilic heads facing away from each other. So we have the hydrophilic head on the facing the outside of the cell, and the hydrophilic head of the other layer going to be facing the inside of the cell. And then the hydrophobic tails are facing each other. So it creates this kind of this bilayer formation. And the phospholipids are going to be the main component of our plasma membrane. So it's going to be majority phospholipid. And as I stated before, because we have this hydrophobic region and this hydrophilic region, we call it an amphithetic molecule. So it basically means that it consists of both of these types of molecules, so hydrophobic and hydrophilic. And the phospholipid bilayer can exist as a stable boundary between the two aqueous compartments. All right. So let's talk about the cell membrane or the cellular membranes. So we like to think of it as a fluid mosaic model. So that basically means that there are, of course, the amphithetic phospholipid bilayer with their hydrophilic portions protruding. And there's proteins that are embedded within this bilayer. So those membrane proteins are going to add to its fluid mosaic description for this particular plasma membrane. So it states that the membrane is a mosaic of these protein molecules that are kind of bobbing in a fluid bilayer of phospholipids. So groups of the certain proteins or certain lipids may associate in long-lasting specialized patches. So we'll see within the membrane, there's, all of these different proteins, and it's going to, these proteins have different functions, and we'll talk about those functions. So here's a good picture of a plasma membrane, or I should say the fluid mosaic model, because it includes the phospholipid bilayer. So you can see the hydrophilic heads facing outside and inside the cell, and then the hydrophobic tails facing each other. Another thing that you'll notice, hey, look at that. This is cholesterol. So cholesterol, we talked about as a steroid. So it's a lipid, and this cholesterol is going to help stabilize the plasma membrane. We have these big proteins. So these proteins, we refer to them as integral proteins. So you can see that they are throughout the plasma membrane. We might have peripheral proteins on the inside of the periphery of the plasma membrane. We might have glycolipids sticking out of the plasma membrane. We might have glycoproteins that compose carbohydrates. We might have single carbohydrate chains attached. So all of these are creating this fluid mosaic model to our plasma membrane. So let's take a closer look at the fluidity. So we have most of the lipids and some proteins in the membrane can shift about sideways. So that's because we have the phospholipids, they're going to be moving. Basically, it's just the hydrophilic heads are kind of attracted to each other. Hydrophobic tails are attracted to each other. So it creates this kind of movement pattern. The proteins embedded, they will move around as well, but they move at a slower pace. Some proteins may move at a directed manner, others may be anchored in place, and then some simply drift in the membrane. So, for example, if we were to merge two cells together, the plasma membranes would merge together. And at first, the plasma membranes would kind of look like, basically the proteins are of one side of the membrane are all together, the proteins on the other side are all together. But then over time, they kind of mix together because this is a fluid mosaic model. So it's fluid, it's going to be moving. These plasma proteins within the membrane do kind of move around and mix. Some other components of the plasma membrane, as it cools, if the temperature does cool, then the membrane switch from a more fluid state to a solid state. So this is going to be, important because as the membrane, if it solidifies, then it's going to basically cause the cell to break apart. So the temperature at which a membrane solidifies depends on the type of lipids it contains. So our lipids are going to be rich in phospholipids with unsaturated hydrocarbon tails. And this is going to allow our plasma membranes to remain fluid even if the temperature drops. So we don't want a solid plasma membrane because that's going to make it more likely to rupture. Okay. Having this fluidity to it is going to allow for movement to take place and so forth. And so that's because of those hydrocarbon tails that are embedded in the hydrophobic area of the plasma or the phospholipids. So it helps keep the fluid nature of our plasma membrane despite drops in temperature. Further, we have cholesterol. So I showed you the little cholesterol embedded within the phospholipid bilayer. So that steroid can help maintain the fluidity at different temperatures. At warm temperatures, the cholesterol helps prevent the phospholipids from moving too much. And then at cool temperatures, it helps keep the phospholipids from packing tightly together. So overall, that cholesterol is going to make sure that the fluid nature of our fluid mosaic model is working properly. So it adds to the stability of the plasma membrane. Okay. So here's an example of what would happen if we didn't have cholesterol. So you could see that if it's too fluid, you end up with these little breaks in our phospholipid bilayer. So that could be, a big problem. Certain things could enter the cell that we don't want it to, or leave the cell if we don't want it to. But we also don't want it too tightly packed together because then certain things can't move through it, and it makes it so that the plasma membrane is going to be so tightly packed together that it's going to be hard for any movement to take place. So that cholesterol is going to help stabilize, okay? So that if the temperature cools down or raises slightly, we maintain this constant kind of situation with the plasma membrane. All right, so let's take a closer look at the proteins that are embedded within the plasma membrane. So I already talked about how there's different types of proteins embedded. So some of them are kind of like within the plasma membrane, some are on the peripheral, and so forth. So we can just basically classify them as integral proteins that penetrate the hydrophobic interior, as well as peripheral proteins that are kind of on the surface of the membrane. So each of these proteins, they help provide a specific function to the membrane. So the integral proteins, they're the ones that will be embedded within the membrane. So They can be called transmembrane proteins because they're within the membrane. And then the hydrophobic regions of the integral protein consist of one or more stretches of nonpolar amino acids. Okay, so that's basically going back to how you can end up having a protein within the hydrophobic area of the membrane. So here's an example of our transmembrane or integral protein. So you can see how we have basically the area that is going to be more nonpolar. It can be that's the part that's going to be facing the edge of the hydrophobic region, and then you might have the more polar areas towards the middle that will open it up from either side. So there's different types or different functions of these membrane proteins. So I'm just going to go over some of the functions here. So some of them are involved with transport. So you can see that this transmembrane or integral protein is going to allow certain things to enter the cell through the little channel that the transport protein is providing. We might have enzymatic activity take place. So we can see how in these particular integral proteins, there are receptor sites on the interior part of the protein facing the inside of the cell, and it's providing a active site for the substrate to bind to. They might be involved with communication. So we have this signal transduction take place where we have a receptor site on our integral protein, and that's going to allow a signal molecule to be received and cause a change in that protein structure. We might have cell-to-cell recognition. So this is also another type of like communication type protein. So we might have a receptor site of a protein that might have some sort of, I don't know, like some sort of carbohydrate side chain sticking out of it. And that's going to allow it to bind to a nearby cell. We might have intracellular joining, so you can see how the receptor sites of both of these transmembrane proteins are going to be providing maybe a gap junction or even a tight junction between the two cells. And then we can also have attachment of the cytoskeleton and the extracellular matrix. So you can see within this protein that's embedded within the membrane, you see the intermediate filaments, or no, these look like microfilaments attached to the protein itself. And then we also have the extracellular matrix that's attached to the outside of that protein. Okay. So what are the roles of carbohydrates in cell-to-cell recognition? We need to have cells be able to communicate with each other and recognize each other. So they do that by recognizing certain carbohydrates that are embedded on the plasma membranes, on that extracellular surface of the plasma membrane. So those carbohydrates can be covalently bonded to lipids. So they end up forming glycolipids, or more commonly the glycoproteins, which are the carbohydrates attached to the proteins. The carbohydrates on the outside of the membrane can vary amongst different species, even individuals. So, you know, they're very specific. Now, how do we make a plasma membrane? So we know that the plasma membrane is this phospholipid bilayer, but where does it come from? Well, it all has to do with that endomembrane system. So remember how we have this endoplasmic reticulum, and that endoplasmic reticulum is made of a lipid bilayer. So when it pinches off to form a transport vesicle, that transport vesicle is going to merge with the Golgi apparatus. So that kind of makes the Golgi apparatus a little bit bigger. It's got some extra lipid bilayer attached to it. And then same with the lipid, the Golgi apparatus will pinch off its own transport vesicle. So that transport vesicle is going to then merge with the plasma membrane. And so now we're adding extra phospholipid bilayer to the plasma membrane. So we're expanding it. Okay, the opposite can occur if we have some nutrient entering the cell. So that will make the plasma membrane shrink a little bit as it pinches off, and the transport vesicle might enter the cell, going where it needs to go. So you can see how the lipid bilayer is constantly, expanding and contracting based on, things that are leaving the cell, causing the plasma membrane to expand, or entering the cell, causing it to contract or become smaller. Let's see. So the plasma membranes are selectively permeable. So we already know that only, certain things can enter the cell and leave the cell. And the way that that happens is that it depends on the substance. Okay, so hydrophobic or non-polar molecules, they can dissolve in the lipid bilayer, and so they end up crossing the membrane pretty easily. Polar molecules, on the other hand, do not cross very easily. So they need to enter the cell usually through some sort of protein that's embedded in the plasma membrane. So I talked to you already about those transport proteins. So they create like a channel that allows the passage of of the polar hydrophilic substances across the membrane. We also have specialized transport proteins that allow water to enter and leave the cell easily, and those are called aquaporins. Water molecules can pass through the membrane, but it's a little slower. So the aquaporins help water move through a lot faster, even though it's a polar molecule, so it doesn't cross that easily. And then we can have carrier proteins, so they bind to certain molecules, and they end up changing the shape of that protein that's in the membrane, and that's going to help open it up so that it can shuttle certain molecules through. All right, so that's it for this particular video.